Hi, welcome to the second part of risk assessment. We're on risk characterization. Quantitative considerations in risk assessment include dose response assessment, exposure assessment, variation in susceptibility, and characterization of uncertainty. For dose response assessment, varying approaches have been proposed for threshold versus non-threshold endpoints. Traditionally, threshold approaches have been applied for assessment of non-cancer endpoints, and non-threshold approaches have been used for cancer endpoints. Dose response assessment. The fundamental basis of the quantitative relationships between exposure to an agent and the incidence of an adverse response is the dose response assessment. Analysis of dose response relationships must start with determination of the critical effects that will be evaluated quantitatively. It is customary to choose the data sets with adverse effects occurring at the lowest levels of exposure. The critical adverse effect is defined as the significant adverse biological effect that occurs at the lowest exposure level. Approaches for characterizing threshold dose-response relationships include identification of NOELs or LOELs, that's NOAEL or LOAEL, which corresponds to the no or lowest observed adverse effect levels. In general, animal bioassays are constructed with sufficient numbers of animals to detect low-level biological responses at the 10% response range. Significance usually refers to both biological and statistical criteria and is dependent on the number of dose levels tested, the number of animals tested at each dose, and the background incidence of the adverse response in non-exposed control groups. The NOAEL should not be perceived as risk-free. As was previously described, approaches for characterizing dose-response relationships include identification of effect levels such as the LD50 or the dose that produces 50% lethality, the LC50, which is the concentration producing 50% lethality, and the ED10, which is the dose producing a 10% response, in addition to the NOELs, NOAELs. So NOELs traditionally have served as the basis for risk assessment calculations, such as reference doses and acceptable daily intake values. Reference doses, RFDs, and reference concentrations, RFCs, are estimates of daily exposure to an agent that is assumed to be without adverse health impact on the human population. Acceptable daily intake, or ADI, values may be defined as the daily intake of a chemical during an entire lifetime that appears to be without appreciable risk on the basis of all known facts at that time. Reference doses and ADI values are typically calculated from NOEL values by dividing uncertainty, or UF, and or modifying factors, which are also abbreviated as MF. So the equations are listed here. RFD, or the reference dose, equals the NOAEL over the uncertainty times modifying factors. And the ADI, or the acceptable daily intake value, equals the NOEL divided by the uncertainty factor times the modifying factor. In principle, dividing by these factors allows for interspecies, animal to human, and intraspecies, human to human variability with default values of 10 each. An additional uncertainty factor can be used to account for experimental inadequacies. For example, to extrapolate from short exposure duration studies to a situation more relevant for a chronic study to account for inadequate number of animals, or to describe other experimental limitations. If only a LOAEL value is available, an additional tenfold factor is commonly used to arrive at a value more comparable to a NOAEL. Traditionally, a safety factor of 100 was used for RFD calculations to extrapolate from a well-conducted animal bioassay, a tenfold factor animal to human, and to account for human variability in response, a tenfold human to human variability factor. Modifying factors can be used to adjust the uncertainty factors if data on mechanisms, pharmacokinetics, or the relevance of the animal response to human risk justify such a modification. Recent efforts have focused on using data-derived factors to replace the tenfold uncertainty factors traditionally used in calculating RFDs and ADIs. Such efforts have included reviewing the human pharmacologic literature from published clinical trials and developing human variability databases for a large range of exposures and clinical conditions. Intra- and interspecies uncertainty factors have two components, toxicokinetics and toxicodynamic aspects. In this image, you can see these distinctions. A key advantage of this approach is that it provides a structure for incorporating scientific information on specific aspects of the overall toxicologic process into the reference dose calculations. Thus, relevant data can replace a portion of the overall uncertainty surrounding these extrapolations. The NOAEL approach has been criticized on several points, including the following. Number one, the NOAEL must, by definition, be one of the experimental doses tested. Two, once this dose is identified, the rest of the dose response curve is ignored. Three, experiments that test fewer animals result in larger NOAELs and thus larger reference doses, rewarding testing procedures that produce less certain rather than more certain NOAEL values. And four, the NOAEL approach does not identify the actual responses at the NOAEL and varies with experimental design, leading to the setting of regulatory limits at varying levels of risk. Because of these limitations, an alternative to the NOAEL approach, the benchmark dose, or the BMD method, was proposed. In this approach, the dose response is modeled in the lower confidence bound for a dose at a specified response level, or a benchmark dose, BMR, is calculated. The BMR usually is specified at 1, 5, or 10%. The BMDX, with an X representing the percent benchmark response, is used as an alternative to the NOAEL value for the reference dose calculations. Thus, the RFD would be equal to RFD equals BMDX divided by the uncertainty factor times modifying factors. The proposed values to be used for the uncertainty factors and modifying factors for BMDs can range from the same factors used for the NOAEL to lower values resulting from increased confidence in the response level and increased recognition of experimental variability resulting from the use of a lower confidence bound on the dose. The advantages of the benchmark dose approach can include one, the ability to take into account the full dose response curve as opposed to focusing on a single test dose, as is done in the NOAEL approach. Two, the inclusion of a measure of variability or a confidence limit. Three, the use of responses within the experimental range versus extrapolation of responses to low doses not tested experimentally. And four, the use of a consistent benchmark response level for RFD calculations across studies. Obviously, limitations in the animal bioassays in regard to minimal test doses for evaluation, shallow dose responses, and the use of study designs with widely spaced test doses will limit the utility of these assays for any type of quantitative assessments, whether NOAEL or BMD-based approaches. Non-threshold approaches. As the image shows, numerous dose response curves can be proposed in the low dose region of the dose response curve. That's down here, if a threshold assumption has not been made. Because the risk assessor generally needs to extrapolate beyond the region of the dose response curve for which experimentally observed data is available, the choice of models to generate curves in this region has received a lot of attention. So this is the possible curves that we can generate here. For non-threshold responses, methods of dose response assessments have also utilized models for extrapolation 10 to the -4 to 10 to the -6 risk levels at very low doses, far below the biologically observed response range and far below the effect levels evaluated for threshold responses. Two general types of dose response models exist: statistical or probability distribution models and mechanistic models. So this is an example of a dose response curve. The statistical significance of these responses is indicated with the star symbol. So that includes this, and this one. The first one is F. The threshold dose shown by a T, which is down here, This is the dose below which no change in biological response occurs. Point E right here is the highest non-statistically significant response. So this corresponds to the NOAEL. Point F, which is the first statistically significant point, corresponds to the LOAEL. Curves A to D show the possible options for extrapolating below point E on the dose response curve. The distribution models are based on the assumption that each individual has a tolerance level for a test agent and that response level is a variable that follows a specific probability distribution function. These responses can be modeled by using a cumulative dose response function. However, extrapolation of the experimental data from 50% response levels to a safe, acceptable, or de minimis level of exposure, for example, 1 in a million risk above background, illustrates the huge gap between scientific observations and highly protective risk limits, sometimes called virtually safe doses, or those corresponding to a 95% upper confidence limit on adverse response rates. Models derive from mechanistic assumptions. This modeling approach designs a mathematical equation to describe dose-response relationships that are consistent with postulated biological mechanisms of response. These models are based on the idea that a response or a toxic effect, in particular biological unit, animal or human, is the result of the random occurrence of one or more biological events or stochastic events. A series of hit models for cancer modeling in which a hit is defined as a critical cellular event that must occur before a toxic effect is produced assume that one, an infinitely large number of targets exists, for example, in the DNA. Two, the organism responds with a toxic response only after a minimum number of targets has been modified. Three, a critical target is altered if a sufficient number of hits occurs. And four, the probability of a hit in the low dose range of the dose response curve is proportional to the dose of the toxicant. The simplest mechanistic model is a one-hit or a one-stage linear model in which only one hit or critical cellular interaction is required for a cell to be altered. As theories of cancer have grown in complexity, multi-hit models have been developed that can describe hypothesized single-target multi-hit events as well as multi-target multi-hit events in carcinogenesis. Toxicologic enhancements of the models. Three exemplary areas of research that have improved the models used in risk extrapolation are time to tumor information, physiologically based toxicokinetic modeling, and biologically based dose response modeling. The biologically based dose response BBDR model is intended to make the generalized mechanistic models discussed in the previous section more clearly reflect the specific biological processes. Measured rates are incorporated into the mechanistic equations to replace default or computer-generated values. The development of biologically based dose response models for endpoints other than cancer has been limited. However, several approaches have been explored in developmental toxicity that use cell cycle kinetics, enzyme activity, litter effects, and the cytotoxicity as critical endpoints. Unfortunately, the lack of specific quantitative biological information for most toxicants and most endpoints limits the study and utilization of these models. One key challenge for toxicologists doing risk assessments will be interpretation and linking of observations from highly sensitive molecular and genome-based methods with the overall process of toxicity. The basic need for linkage of observations was highlighted in early biomarker work. Biomarkers of early effects, such as frank clinical pathology, arise as a function of exposure, response, and time. Early, subtle, and possibly reversible effects generally can be distinguished from irreversible disease states. The challenge for the interpretation of early and highly sensitive responses, or biomarkers, is made clear in the analysis of data from gene expression arrays. Because our relatively routine ability to monitor gene responses, up to tens of thousands of them simultaneously, has grown exponentially in the last 5 years, the need for toxicologists to interpret such observations for risk assessment and for the overall process of toxicity has been magnified with equal or greater intensity. For microarray data, each gene response or cluster of genes can have its own pattern of response, peak, and overall duration of response. Microarray analysis for risk assessment requires sophisticated analyses. Because of the vast number of measured responses with gene expression arrays, Pattern analysis techniques are being used. The extensive databases across chemical classes, pathological conditions, and stages of disease progression that are essential for these analyses are being developed. Exposure assessment. The primary objectives of exposure assessment are to determine the source, type, magnitude, and duration of contact with the agent of interest. Obviously, a key element of the risk assessment process, hazard does not occur in the absence of exposure. However, exposure data are frequently identified as the key area of uncertainty in overall risk determination. The primary goal of using exposure information in quantitative risk assessment is not only to determine the type and amount of total exposure, but also to find out specifically how much may be reaching target tissues. A key step in making an exposure assessment is determining which exposure pathways are relevant for the risk scenario under development. The subsequent steps entail quantitating each pathway identified as a potentially relevant exposure and then summarizing those pathway-specific exposures for the calculation of the overall exposure. Additional considerations for exposure assessments include how time, duration of exposures are evaluated in risk assessments. In general, estimates for cancer risk uses averages over a lifetime. In a few cases, short-term exposure limits, or STELs, are required, and characterization of brief but high levels of exposure is required. In these cases, exposures are not averaged over a lifetime. With developmental toxicity, a single exposure can be sufficient to produce an adverse developmental effect. Thus, daily doses rather than lifetime weighted averages are used. Variation and susceptibility. Toxicology has been slow to recognize the marked variation among humans. Generally, assay results in toxicokinetic modeling utilize means and standard deviations to measure variation, or even standard errors of the mean, to make the range as small as possible. Outliers are seldom investigated. Thus, it is important to know whether and how one patient might be at higher risk than others. When investigators focus on the most susceptible individuals, there may be a better chance of recognizing and figuring out what the underlying mechanisms of toxicity are. Host factors that influence susceptibility to environmental exposures include genetic traits, sex, age, pre-existing disease, behavioral traits. Some important ones are smoking, chronic ethanol ingestion, coexisting exposures, medications, vitamins, and protective measures. Genetic studies are of two kinds. One, investigations into the effects of chemicals and radiation in the genes and chromosomes. And two, ecogenetic studies, which identify inherited variation and susceptibility, or predisposition and resistance to specific exposures, ranging across pharmaceuticals, pesticides, inhaled pollutants, foods, food additives, sensory stimuli, allergic and sensitizing agents, and infectious agents. Inherited variation and susceptibility has been demonstrated for all these kinds of external agents. The Toxicology Data Network, or ToxNet, from the National Library of Medicine provides access to databases on toxicology, hazardous chemicals, and related areas. These information sources vary in terms of the included level of assessment, ranging from listings of scientific references without comment to extensive peer-reviewed risk assessment information. The World Health Organization provides chemical-specific information through the International Program on Chemical Safety and documents health and safety documents. The International Agency for Research on Cancer, IARC, provides data on specific classes of carcinogens as well as individual agents. The National Institutes for Environmental Health Sciences, NIEHS, National Toxicology Program, provides technical reports on the compounds tested as a part of this national program, and its reports on carcinogenesis, or carcinogens. Also provides carcinogen-specific information on their website. Risk perception and comparative analysis of risk. Individuals respond very differently to information about hazardous situations and products, as do communities and whole societies. Understanding these behavioral responses is critical in stimulating constructive risk communication and evaluating potential risk management options. In a classic study, students, League of Women Voters members, active club members, and scientific experts were asked to rank 30 activities or agents in order of their annual contribution to deaths. Club members ranked pesticides, spray cans, and nuclear power as safer than did other laypersons. Students ranked contraceptives and food preservatives as riskier and mountain climbing as safer than did others. Experts ranked electric power, surgery, swimming, and x-rays as more risky and nuclear power and police work as less risky than did laypersons. There were also group differences in perceptions of risk from chemicals among toxicologists correlated with their employment in industry, academia, or in the government. Psychological factors such as dread, perceived uncontrollability, and involuntary exposure interact with factors that represent the extent to which hazard is familiar, observable, and essential for daily living. Public demand for government regulations often focuses on involuntary exposures, especially in the food supply, drinking water, and the air, and unfamiliar hazards such as radioactive waste, electromagnetic fields, asbestos insulation, and genetically modified crops and foods. Many people respond very negatively when they perceive that information about hazards or even about new technologies without reported hazards has been withheld by the manufacturers. Genetically modified foods or government agencies, like HIV-contaminated blood transfusions in the 1980s, the extent of hazardous chemical or radioactive waste. Most people regularly compare the risks of alternative activities on the job, in recreational pursuits, and in interpersonal interactions and in investments. Determining how to best conduct comparative risk analysis has proved difficult because of the great variety of health and environmental benefits, the gross uncertainties of dollar estimates of benefits and costs, and the different distribution of benefits and costs across the population. To summarize, the National Research Council and Risk Commission frameworks for risk assessment and risk management provide a consistent, data-based approach for evaluating risks and taking action to reduce risks. The objectives of risk assessments vary with issues, risk management needs, and statutory requirements. However, the frameworks are sufficiently flexible to address these variations or these various objectives and accommodate new knowledge while also providing guidance for priority setting in industry, environmental organizations, and governmental, regulatory, and public health agencies. Toxicology, epidemiology, exposure assessment, and clinical observations can be linked with biomarkers, cross-species investigations of mechanisms of effects, and systemic approaches to risk assessment, risk communication, and risk management. Advances in toxicology are certain to improve the quality of risk assessments for a broad array of health endpoints as scientific findings substitute data for assumptions and help describe and model uncertainty more credibly. Thank you for watching.